Dear students, today I am just going to tell you about the megasporogenesis, pollination and development of embryo. In the previous video, I have already spoken about microsporogenesis. Now question comes, what is megasporogenesis? Megasporogenesis, like macrosporogenesis, it is again the differentiation of megaspore mother cell into megaspores. Remember in megasporogenesis, the four megaspores which are being formed, out of them three disintegrates and one functional megaspore is there. So one megaspore develops into embryo sac, or we also call it a female gametophyte. Megasporogenesis occurs inside the megasporangium, and megasporangium is also known as the ovule. Don't get confused with that. Each megasporangium or ovule has got three main parts. Number one, that is the integument. Second is the nucellus, and the third one is the funicle. Integument forms the protective layer. Then nucellus, it is the nutritive layer from which the female gametophyte develops, and the funiculus, it is the stalk of the ovule by which it remains attached to the ovary. Here you can see the formation of the embryo sac or the female gametophyte. The megaspore mother cell, it just undergoes meiosis, forms the four megaspores, three disintegrates, one remain functional, and this one functional megaspore, it undergoes three successive mitotic divisions. Now, as a result of three mitotic divisions, 8 nucleated stage is being formed, and all of them, these nuclei, they arrange themselves in a proper way. You can just see the three nuclei at the macropylar end, the three nuclei at the chalazial, this is the chalazial end opposite to the macropylar region, and the two nuclei from each pole, they come in the center. As a result, 8 nuclei, these two nuclei, they remain close to each other and they form a single cell like structure cell, we call it a central cell. So it is being asked in the examination that the embryo sac or the female gametophyte is how many nucleated and how many celled. Then after the formation of the female gametophyte, then the pollination we should come to the pollination is essential now, because the fertilization has to take place. So pollination is of two types, you can say, the self pollination and the cross pollination. I think you are aware with the pollination term. So the transfer of the pollen grains from the anther to the stigma. In self-pollination is also known as autogamy, cross-pollination is known as allogamy. Now this cross-pollination is of two types again, geitonogamy as well as the xenogamy. Geitonogamy means generally when the pollens of one plant falls on the stigma of the flowers of the same plant. So there is no genetic variation. Whereas xenogamy, here the plants of the same species, different plants of the same species or the different species could be there. Flowers now. When we speak about the pollination, then a question comes to our mind. Then in some cases the flowers remain closed, and in some the flowers remain open. What do we call these closed flowers as? The term is clistogamous and the chasmogamous. Again, the differences could be asked between these two. Clistogamous, here, like you have studied about the garden pea in class 10th, where the flowers remain completely closed. The male part and the female part, they are not exposed to the environment. It completely shows self-pollination, true breeding plants, clistogamous. Whereas the chasmogamous, here they can show self-pollination as well as the cross-pollination both. But there are some plants which possesses both chasmogamous as well as clistogamous flowers. I'll give you the example like viola, like oxalis, and so on. Now in pollination, but still I have told you about the pollination, there are sometimes what happens, the pollens fail to germinate on the stigma of the same flower, especially in the bisexual flowers normally it occurs. Why it is so? The flowers are bisexual, they are chasmogamous, but the pollen grains are not able to germinate. The reason is because they prefer the cross pollination for genetic variability. So in that them these case in these plants, the outbreeding devices are being developed. First of all, let us understand why it happens, why they do not germinate. There are certain reasons for that. Either the pollen as well as the stigma, they do not mature at the same time, or their positions could be different. Then the third, they may be showing the genetical incompatibility, which is known as the self-incompatibility, where due to certain factors, the stigma is not allowing the pollen grains to germinate over it, and or you can say the presence of the unisexual flowers. Here in this figure, you can just see how the pollination is taking place. It is showing the autogamy as well as the geitonogamy. Once the fertilization is over, now pollen grains, when pollination occurs, then pollen tube will be formed, and pollen tube will lead to the formation of the will lead to, you can say, fertilization. Here in this type figure, you can normally see a pollen tube here. This pollen tube consists of now two male nuclei, the red ones, and the second diagram here you can see the pollen tube has entered through the micropyle, and these two nucleus, out of one nucleus, one fuses with the egg, and the other one fuses with the two polar nuclei, which have come from the two poles and aligned themselves in the center. Then you can see the triple fusion. In this case, here you see the triple fusion has taken place. So you can say syngamy is the fusion of male and female gamete here to form zygote, and the triple fusion means the fusion of two polar nuclei with one of the nucleus, resulting in triploid nucleus. This together is termed as the double fertilization. As a result of the double fertilization, all these nuclei, antipodal cells, synergids, they all disintegrates. Only surviving one is the PEN, the primary endosperm nucleus, as well as the zygote. Zygote develops into the embryo, whereas primary endosperm nucleus develops into the nutritive tissue known as the endosperm. We speak about the endosperm. That endosperm, you remember that it is the nutritive tissue which nourishes the developing embryo in the embryo sac. Now the division of the formation of the embryo endosperm is prior to the you can say formation of the zygote. The primary endosperm nucleus or the triploid nucleus, it divides. There are three ways of dividing of the formation of the endosperm. Number first is the nuclear type. Here this is a nuclear type, this is a cellular type, and this one is the halobial type. In nuclear type, you can see the nucleus is dividing, which is not for being followed by the division of the cytoplasm. So you can see multinucleated state, and here the cellular division is also taking, the division of cytoplasm is also taking place in the second case. So cellular nucleus, cellular endosperm, and a third case, which is generally, which is also seen in the coconut. The question is being asked many times about that. The coconut water, coconut kernel, what is that? Which type of endosperm it is? So it is showing what is the both type. You can see the nuclear as well as the cellular type. You can see the first nuclear division takes place, then followed by the cytokines. Two cells can be seen again in these two cells. One is, both of them, they are going the free nuclear division, or one can show the free nuclear division and other can show the cellular division. So we call this type of endosperm as halobial type. So coconut water is free nuclear, whereas coconut kernel is the cellular endosperm. Once this endosperm is over, now here what happens to the zygote? Let us see. Is the zygote will develop into the embryo. The zygote divides by transverse division into two cells. You can see here the one is the basal cell and the other one is the embryonal cell. This is the embryonal, the basal cell. It divides, redivides, and it forms the row of cells. Here in this diagram you can just see this row of cells, which is known as a suspensor, followed by this one is the terminal cell of the suspensor. Here it swells up and it absorbs the nourishment nutrients from the endosperm. It get embedded into the endosperm and provide the nutrition through the suspensor. It goes to this zygote. Embryonal cell, you can see embryonal cell divides, redivides, and it forms the globular embryo, which you can see here. Then further development takes place. What happens? You can see the heart shaped embryo is being formed. Now the endosperm is finished. Its whole nutrient is being absorbed by the embryo, and food gets stored in these two limbs. You can see the cotyledons. Here in the last diagram, you can see the mature embryo with the two cotyledons. In the center, you can see the embryonal axis, the disintegrating suspensor, and the one side radical, and the center here you can see the plumule. After the formation of embryo, now the ovule or megasporangium, which I used a term earlier, it get converted into seed. So what is seed now? It is fertilized mature ovule is known as the seed. Different type of seeds can be seen in this figure. Now seeds could be of two types. Number one, albuminous seeds. Second, the non-albuminous seeds. Albuminous seeds, they are also known as endospermic seeds, whereas non-albuminous seeds are called X-albuminous seeds. X-albuminous seeds may without the endosperm. So remember that all the monocots mostly they have got they are the endospermic seeds because they have the endosperm and which contain the food, and the dicot seeds are mostly non-albuminous, or you can say X-albuminous endospermic seeds. The name has been given over here. Then castor is being mentioned in this slide, which is a dicot seed, but being dicot still, it is an endospermic seed. You can see the diagram of the endospermic as well as exendospermic seed, and you can say this is the bean seed and this one is the maize seed. This portion is the endosperm, and here the endosperm is completely finished off, and we call this portion as the cotyledons. Now seeds when they are being formed, then they are being dispersed by various methods also, either by the animals or by the wind or by the water. Different ways of dissemination of seeds are there, but some of the seeds they do not germinate even under favorable conditions. They undergo a resting stage in their life. We call it the as seed dormancy. Seed dormancy means when they fail to germinate. Why do they fail? The reason is because either the embryo present inside it may not have developed completely, so they require the period for its growth also. So when the this advantage, what could be more advantage of this seed dormancy? Seed dormancy helps the farmers to store the seeds. They could be transported easily from one place to another. Seed dormancy, after now reasons of the seed dormancy, also the seed dormancy is caused by various factors such as the hard seed coat, the presence of certain chemicals like abscisic acid on the seed coat, as well as the rudimentary embryo. So it can be broken by various means, either by rubbing them or by certain treatments such as treating with them with the acids or soaking them in the water for some time and then sowing them. Next comes the fruit. Now seed is ready. Now seeds are born especially in the angiosperms, especially inside the ovary. Whole ovary now, the mature ovary, the ovary which contain the fertilized seeds especially, that is known as the fruit. Fruits are of generally two types. Number one, true fruit as well as the false fruit. True fruits that derive from the ovary, that fruits in which are formed from the ovary only, they are called the true fruits. And fruits which are derived from the parts other than the ovary, like thalamus, like sepals, petals, they are also contributing whole inflorescence. They're contributing in the formation of fruit. We call them the false fruit. So apple is here. The thalamus, fleshy thalamus, is converted into fruit. Now, besides this, there's one more category of the fruit known as the parthenocarpic fruits, like banana, which is seedless. So these fruits are known as the parthenocarpic fruits. Next is the apomixis. This is again a phenomena now which is used by the farmers nowadays. This is the phenomena where you can develop the seed. See, when we say about the seed is the result of fertilization. But now we are developing the seeds without undergoing sexual reproduction fertilization. Then this is known as the apomixis. And apomixis, you can say it is an asexual method of reproduction. This apomixis play a very important role for the farmers. This can be stored also. The diploid tissues can be converted into seeds, or they can be converted into seeds. In fact, then here you can see the figure shows the Mendel sort of experiment, the genetic experiment. Here you can see in this way here the hybrid which has been formed in the F1 generation. What happens with the good characters or hybrid vigor? You can see heterosis means the good characters, more production. This plant now, sometime whatever, if we just store the seeds of this plant and propagate them, grow them again and again, what you'll find, then you'll see the hybrid vigor or heterosis or the more production will be affected. The depression can be seen in the production over here. So if you remember the Mendel's crosses where the after generation shows the ratio 75% and 25%, the same way you can see that all the plants will not be developing good amount of production or the seeds. But if what happens in this hybrid only, the hybrid seeds are being made apomicted. Okay, and then they are being stored and grown again and again. What will happen? All that the production will be, or you can see here, all the plants are of same type. The production will not be affected. So it is a useful event for the farmers, you can say. Then next last is the polyembryony. Polyembryony is a phenomena where more than one embryo is especially seen in case of certain plants such as belonging to mango, such as citrus and others. More than one embryo, I am saying, is being seen. Now, what is there here? How the more than one embryo can be formed? Just you can say either by some diploid tissues such as nucellus or integument. They move inside the embryo sac and start developing into the embryo. Now again, this is of advantage for to the farmers in giving the good yield also. The differences between the apomixis as well as polyembryonic is being shown. This is all in this video. I think I have tried my best to complete all the topics of this lesson within a short span of time, and hope you will enjoy it. Best of luck. In case if you have some any queries, please subscribe it as well as put the comments wherever you have any doubt.